Okay, and so I am I guess by training, an environmental engineer and an anaerobic microbiologist. So certainly from my perspective, I really come from this world, from the anaerobes perspective. And I guess early on in kind of my postdoc training, with Hector actually, I started to meet automation. And so ever since then, I've really been inspired by it and bringing it into a lot of the work that we do. And so as a lab, we are exclusively kind of focused on industrial microbiome engineering, trying to think about conversion of waste to chemicals. So that's really what motivates a lot of the efforts we're doing. Okay, so there's about 5 billion tons of food waste or organic waste produced every year. So that's a huge, huge number when we think about the volume of that feedstock. And what we're really interested in doing is thinking about how all these diverse types of wastes can be used for chemical and fuel manufacture, right? And so when we think about making fuels and chemicals from waste, of course, anaerobes are kind of some of the key stars. And one technology in particular that's used exclusively at scale already is called anaerobic digestion. And so anaerobic digestion is a really nice technology. We like it because we say it has a big mouth, right? It can use a wide variety of different types of feedstocks and convert them to primarily one end product currently, right? So it's good at making biogas. You can take out the CO2, produce methane, which is called renewable natural gas, which is great. But our lab is really thinking about what else could anaerobic fermentation more broadly as a platform do. So can we use this platform to move beyond biogas to make our chemicals and fuels? And so historically, when we thought about engineering microbial communities to make products beyond biogas, oftentimes you get pushback. Microbiomes are too complicated. Tools are very limited. Everything's very empirical, trial and error, really difficult. And so broadly, what our lab's been focused on is kind of opening up this black box, understanding different organisms, particularly their functions and how they interact in these systems, but more importantly, trying to develop more bottom-up approaches for how microbiomes form and how that formation can control their function. And of course, can we manipulate them genetically to introduce new pathways, new enzymes into these organisms to actually be able to make products that one can't just naturally select for. And so a lot of these questions obviously motivate the need for new tools to be able to both study but also manipulate the function of anaerobic microbes as well as microbial community. Okay, and so this is kind of the overarching vision for our lab. We like to take what's called a top-down, bottom-up approach for developing synthetic communities that can be tailored for organic waste to chemical conversion. And so what we do in the lab is we know there's a large diversity of microbes on the planet that already do a lot of the functions that we want. So they're already really good at breaking down lignocellulosic materials, protein materials, waste in general. And so what we do first is we use selection. And so we'll feed a bioreactor with contents from a rumen, for example, cow poop, goat poop, or we'll take sludge from an existing digester and operate a process to select for a microbial community that natively performs a lot of the functions for us already. And so this might be complex polymer degradation to key intermediates, and then conversion of those intermediates to, let's say, a volatile fatty acid or a medium chain fatty acid. And so that's very traditional, that's typical, that's usually what's already done in anaerobic microbiology and anaerobic biotechnology. And so what we've really looked at in our lab for the last 3.5 years is a more bottom-up approach to this. And so what we've been doing is we've been taking those mixed communities and we've been using automation and high throughput isolation approaches to build out what we call a biobank. So what a biobank here is a diverse set of functionally diverse set of microbes that mediate different steps in the process. So this again could be arabinoxylan conversion to lactate or lactate conversion to medium chain fatty acids. And then with that biobank, we use automated design, build, test, learn cycles. From Paul's talk today, we use level 2 automation to be able to build out these synthetic microbial communities, which I'll talk about near the end of my talk today. And so in doing so, we can mix and match the right microbes with the right functional capabilities to be able to convert our complex waste feedstocks into the products that we're interested in. We've also been spending quite a lot of time developing genetic tools with some collaborators for onboarding non-model microbes that we're finding and isolating from these different environments. And so overall, the main motivations for implementing this vision are really, can we boost trimetrics, right? Tighter rates yields of our product of interest from a given waste feedstock? Can we use these synthetic communities not only for industrial biomanufacturing, but really to understand basic questions about microbial ecology, formations of communities, and then use that insight to be able to better develop our processes? And of course, can we make products of higher value? So beyond biogas, can we make, really use anaerobic fermentation and microbial consortia as a platform technology for broadly making fuels and chemicals? Okay, so when we think about what products we want to make using anaerobic fermentation from waste, we like to think of this kind of value volume pyramid, right? So at the base of the pyramid are things globally we use a lot of, right? So in the billions of tons per year. So this would be things like energy molecules, right? At the very top of the pyramid are fine chemicals. So these are things that have a very, very high price, but we only need a really small amount of them. So what we're trying to do is really advance anaerobic digestion from this high volume, low value product, kind of up to the middle of the pyramid, where we're making bulk chemicals, plastics, and polymers. So we still offset a lot of CO2 emissions from feedstocks such as petroleum, but we're also getting a higher value product out of it that really helps drive the economics. Because when you're in the business of turning waste into still a relatively low value product, a chemical of say $5 a kilogram, the margins are quite thin to be able to make an economically viable process. And so we think that this really being able to make a higher value product from waste would really allow the economics to be in the right position to commercialize this. Okay, so when you think about what products to make, obviously everything in chemicals and the petrochemical sector would be a great starting point, and this is where we've started. But certainly, not all of the fuel sector is expected to electrify, so there's still long-haul transportation fuels, sustainable aviation fuels. These are also products we're certainly very interested in making using anaerobic fermentation with waste as a feedstock. And so as a starting point, my lab is focused on a chemical or a class of chemicals called medium-chain oleochemicals, and particularly starting with medium-chain fatty acids. And so these are a particularly good starting point for waste to chemicals manufacturing for a couple reasons. Medium-chain fatty acids are chain length C6 to C12, as you can see here. And they're really used in a wide variety of products. And so you'll find them in nutraceutical products or promotion of your health. I went to the Shoppers Drug Mart, which is a Canadian store you might not be familiar with. And I looked at the ingredients list, and you'll find it in different personal care products, fragrances, these kind of things. One of the biggest markets of medium-chain fatty acids is actually as an antimicrobial. So medium-chain fatty acids display really potent antimicrobial properties. And so the biggest market right now for them is actually as an animal feed additive. And so they're replacing antibiotic usage and livestock cultivation. So that's one thing. They're used in a wide variety of products. Right now, MCFAs come from palm and coconut oil refining. And so as the picture kind of says, there's lots of negative consequences of that environmentally. Large greenhouse gas emissions, deforestation, land use issues. Moreover, though, there's lots of supply chain issues. A lot of the plantation of palm and coconut trees happens overseas, right? And so if crop productions are low that year, there's big supply chain issues and price volatility. And so folks that want to use these MCFAs in other regions suffer the consequences. And so both of these things really motivate, on the demand side, a need for more environmentally friendly MCFA sources that are locally produced. And so as I've already kind of said, MCFAs themselves are a great product to start with, not only because they have a lot of direct uses, but they really feed in and are at the base of the medium-chain oleochemical market broadly, right? So we can take MCFAs, we can reduce them to their corresponding alcohols and alkanes, which have, themselves wide variety of uses industrially, solvents, fuels, things like this. And you can also produce, things like diacids, diols. Diacids are precursors, for example, to nylon. So lots of different derivative products that can be made from MCFAs, either currently by coupling biological processes with catalysis, right? Or of course, something we're very interested in is using metabolic engineering to modify or tweak these microbes that can make these MCFAs using metabolic engineering. And so this really would broaden the size of products that we can make from anaerobic fermentation and really catalyze this biorefinery concept that many of us are very interested in seeing realized. Okay, and so how do we then turn waste into medium chain fatty acids? So here is a very classic anaerobic fermentation food web that many of you are probably familiar with. And so typical anaerobic digestion works by taking your complex biopolymers in the waste, so complex carbohydrates, proteins, lipids, and then breaking them down into their monomers. And then those monomers get fermented into acids and alcohols, which ultimately go to methane, right? Biogas. What we do is at that first step, that selection phase, is we inhibit methane production and we get the accumulation of key intermediates. Initially, lactate and ethanol, right now in literature, are really described as two key ones. As I'll mention in my talk, there's several others that are very important. And ultimately, via the combination of two major groups of microbes, lactic acid bacteria and these chain elongating bacteria, those complex biopolymers, or the carbon in those complex biopolymers, are funneled to the intermediates and then fermented to medium chain fatty acids via a process called chain elongation, which is based on a pathway called reverse beta oxidation. Okay, so this works in the lab great. You can inhibit methanogenesis. We say methanogens are kind of snowflake microbes, so you can keep the pH low, you can bump in a couple toxins, and you can get rid of them quite readily. But even with an undefined microbiome, so an open culture, which is conventionally used, there's still quite some limitations. So with any digestion process, when you're looking at a relatively recalcitrant organic waste feedstock, of course, hydrolysis is a bottleneck. How much of that feedstock can you actually break down and convert into a product that ultimately affects your yield? The other main issue, of course, especially when you're making chemicals, is selectivity. So right now with an undefined microbiome, we can get some MCFAs produced, right? But we also get butyrate produced and acetate and some hydrogen, and we still get some leaky methane production. And so it's not optimal. And ultimately, when we're looking at the economics of the process, how much yield we're getting from that feedstock is really critical. Currently, right now, there's no genetic tools that can be used for these microbes, right? Or these mixed communities. And so you're limited exclusively to C6 and C8 MCFAs. We can't make the C10s and we can't make the C12s. Biology naturally doesn't make those. And so we have a limited product range, not to mention all the other derivative products I mentioned. And one of the biggest problems with any fermentation process, especially any anaerobic fermentation process, is half your feedstock when you put it in goes up in the air as CO2, right? And so one of the main considerations that we've been thinking about as motivation for our future work is how do we keep that CO2 fixed in the process and ending up in our end product. So that's big picture of kind of the lay of the land of making MCFAs. And what we've been thinking about or envisioning is, what would this synthetic community then look like that's taking food waste, for example, or another source of organic waste to chemicals, with our targets being hydrolysis, selectivity, new products, and CO2 loss. So we would have some complex biopolymer. This is saying starch, but we can, you can put all of the mixtures of polymers in there from your waste source. You'd have a lactic acid bacteria that's taking that substrate to lactate, a chain elongating microbe that's taking the lactate to the MCFAs, but they're also emitting the CO2. And then you could couple this with external sources of energy. So that could be taking the dry fraction of your organic waste, for example, gasifying it to a syngas and adding that into your process, or generating hydrogen renewably and adding that into your process, and feeding it to a gas fermentation microbe. We call this process together a mixotrophic anaerobic fermentation. So we're feeding a gaseous feedstock as well as a solid organic feedstock together. And this would enable complete recycling of the carbon to our MCFAs. That obviously ideal scenario, theoretical yields of 100%. And so this is great in grand vision. And when I started my lab, I was like, oh, let's do this right away. But of course, it's taking time to build up to that. And so I'll show you our progress to ultimately charting toward this full carbon recycling community. starting with the stars of the show, these chain elongating microbes, which are at the business end, directly involved in synthesizing medium chain fatty acids. Okay, so chain elongating microbes are actually not new. They've been around, or they were initially discovered back in as early as the 1930s. Clostridium claivri, I'm sure many of you have heard of that organism. It's kind of seen as the model chain elongator. And really in the 2010s, when these open communities started popping up, there were a lot more isolates that have been kind of deposited into culture banks of these chain elongators. And so now, there's about 20 or plus more isolates of chain elongating organisms that you'll find in the literature. And what you'll note is that they are taxonomically diverse, right? So they cover many different families of anaerobes, and that you'll find them in from different sources, right? So claivri was from the mud, Megasphaera is from kind of the rumen. Our favorite Pseudoramobacter lactyliticus is from a tooth infection, right? I don't know if Paul knows that one. He mentioned he works with teeth. And so they're very diverse. And so we have a lot of organisms or chain elongators even off the shelf to start with, right? And so you're building a synthetic community. The question is, what chain elongator do you use, right? Do they have different capabilities, different growth, different phenotypes? How do those phenotypes partition across the different organisms, and what niches do they occupy? Of course, can they be genetically modified? Can we put tools in to actually be able to manipulate their biology? And then do they form stable communities? So lots of questions, even with this handful of chain elongators to start with, right? So when we looked at this initially in the lab, again, about 3.5 years ago, the first thing we thought was, okay, we got to do some automation. We got to be able to screen these anaerobes and higher throughput to be able to collect some data, because many of these microbes, never mind genetic tools, don't even have growth curves for them, right? So how do we do more higher throughput characterization of these organisms? And so if you're going to do anaerobic microbiology with automation, you need a large chamber. And so we had that, and then we started to put our anaerobic equipment inside of it, right? And so this is just a simple but very useful set of equipment we have in the lab. Consisted of a liquid handling robot, incubator, 8-tray incubator, and a plate reader that we've been using to initially characterize these organisms. Here's kind of just a more graphical representation, right? Where we can integrate these and run experiments up to 1,000 parallel conditions, or 1,000 experiments per run. And I say per run, of course, because the run length depends on how slow growing, of course, the anaerobe, right? So usually we're running experiments every week. So one new experiment a week, 1,000 different conditions. I have some videos here. I don't know if these are going to work. I'm just kind of showing the system in a bit more detail. That one works. Other one's not looking like it works. But it's a pretty simple setup. You can see kind of a deck layout here. This is actually some of the experiments I'll talk about later, where we're building synthetic communities that are directly converting pulp. You can actually see here, this is food waste pulp that has a solids content. We have these deep well 96 plates that we're using to store all of our different strain biobank, that we build a plate using a small but mighty 4-channel colony picking robot, or hit picking robot, sorry, that then passes the plate to a biospa, which you can see here, incubates the plate, and depending on the assay we're doing, can also pass it to a plate reader, and all this is done in an automated way. So relatively simple system, lots of lessons learned, certainly about doing practical anaerobic microbiology in an automated system like this. So thinking about how do you minimize contamination, what are optimal plate combinations, how do you minimize evaporation? There was a question about evaporation. So lots of nice lessons learned trying to do anaerobic microbiology for real in an automated system. Oh, there goes the other video. Okay, and so back to our hit list of different chain elongators. And so some of the first things we wanted to do is just systematically screen them, right? What do their growth phenotypes look like on different substrates, different sugars, alcohols, acids, and which organisms in particular produce MCFAs at high concentrations, right? And so here you can just see the system being able to produce nice growth curves for us. We can take our plates and we can measure their chemistry using our GCMS system, and we can get important bioprocess parameters, growth rates, yields, things like this, right? And so one of the first microbes, which is one of the reasons why it's our favorite, although there's a new favorite, which I'll talk about later, is a lactolyticus because in particular, it grows well on lactate, which is, again, one of those key intermediates in chain elongation where everything kind of tends to be funneled to. And it has quite high yields of C6 and C8 medium chain fatty acids, so hexanoic and octanoic acid. The other thing we've been doing is we've been screening a broad range of substrates. As I mentioned, lots of things can go into this chain elongation process. Typically, it's been thought lactate and ethanol are some of the key substrates. From our screening, we found out these organisms are more diverse phenotypically than what's been previously thought. So you can see they like also glycerols are really nice substrates. Up here, you can see a number of them tend to use different types of sugars as well. And so this has been useful for, again, just getting the phenotypic breadth of some of these organisms and understanding their capabilities. One of the other main things, of course, is looking at their toxicity. So we've used the platform to screen how are these microbes tolerant to the MCFAs? As I mentioned, medium chain fatty acids themselves are an antimicrobial, right? And so what you're seeing here is just reductions in growth based on increasing concentration of some of these MCFAs. Here you can see in the blue bars are kind of classic E. coli strains that have been engineered to make MCFAs aerobically. And you can see they kind of pedal out as MCFA concentrations increase, while a lot of these organisms that naturally make MCFAs are a lot more robust to the concentration and higher titers one would expect in a bioreactor. And so you can see here, this is with octanoic acid, concentrations, if you look at the x-axis, dipping down, and decanoic acid, really most of these organisms are quite sensitive to. You know, in the 500 milligrams per liter range, we found some organisms that could grow kind of at that concentration, but you see maybe already just from this environmental toxicity screen, some of the evolutionary constraints on the C8 production that these organisms can do might be just the simple fact that they're quite toxic, right? Okay, so we've been using the platform to screen these organisms. One of the other things that we were doing with my student Ian and Joel is really varying the ratio of media components when growing chain elongators, especially their substrates, for example, lactate as well as acetate, and looking at the consequence of varying those ratios. And one of the really interesting things my student found was that when you have really high concentrations of lactate and low concentrations of acetate, they tend to grow a lot slower. When you add more acetate into the media, which is not actually used as it's not consumed, which is interesting, but when it's present, they tend to grow a lot faster. Even more interesting was this trade-off between growth rate and MCFA chain length. And so as I mentioned, the MCFAs we're really interested in are the longer chain length ones, C6 and C8 over C4. And when the organisms are fed higher ratios of lactate to acetate, what you find is that their product distribution changes and shifts to those longer chain fatty acids, which is really interesting to us. And so with the platform, we were able to really investigate this, and I'll just share some highlights on that right now. But really the main question is, what is explaining this trade-off between growth rate and MCFA yield? That was a really intriguing question for us. Okay, and so this was some work that we've recently put on bioRxiv. So if you want, you can check it out, and it's in review currently. But if you're, this pathway, and I promise this is the only little bit of, metabolism I'll talk more about today. But the way MCFA production works is you start with an electron donor like lactate. It gets converted to acetyl-CoA. Acetate can also be brought into the acetyl-CoA pool, and then 2 acetyl-CoA units are reduced and then subsequently, or condensed, and then subsequently reduced around the reverse beta oxidation cycle. And as you go around the cycle once, you spit out butyrate. If you go around the cycle twice, you spit out hexanoic acid, 3 times octanoic acid. And so one of the main questions we were looking at was, how does lactate versus acetate ratio explain what's happening here? And so like a good anaerobic microbiologist, we thought maybe redox constraints were involved here. And so one of the things we started doing was just some simple biochemistry redox balancing, right? And so when you are oxidizing lactate and you have acetate available, right, you're generating 2 NADH reducing equivalents, and you need 3 reducing equivalents to run the RBO cycle, right? And so what these organisms can do is they can take one of the ferredoxins they produce in this cycle, they can run it through this really unique complex called the RNF complex, generate some energy, and generate the NADH needed to complete this, to balance redox, which is a fundamental thing all anaerobes have to do, right? And they can get some energy out of it as well. And so that's when they're taking lactate and acetate. But the main question is what happens when there's no acetate available, right? So then they have to take 2 moles of lactate to get 2 moles of acetyl-CoA. So the pathway is built on 2 carbon units. You stitch them together, you get butyrate. You need 4 moles of NADH. And so the problem here is that now you have this redox imbalance where you can burn one of your ferredoxins with the hydrogenase, but you then have to run one of your NADHs in reverse through this RNF complex. And that's not theoretically, thermodynamically possible. So you can't do that. And so what these organisms then do to meet that redox constraint is go around the cycle twice to make the MCFAs, right? And so now they have 6 NADHs. You can go around the cycle twice. 3 times 2 is 6. It all balances, right? And so we've plotted this across organisms that do chain elongation to C6 and C8 MCFAs, as well as organisms that seemingly just stick to their lifestyle of making butyrate. And we can come up with these really nice curves where the organisms that make are MCFA producing, right, tend to walk this redox imbalance and increase their cycle depending on the amount of acetate that's available. So you can read about the gory details here. But the trade-off, of course, is that when you're making longer chain products, there's a larger enzyme investment required to actually run the cycle twice. And so this is why they tend to grow slower. So there's this trade-off between a growth penalty and having a higher proteome constraint, having to run the cycle longer versus chain length. So this is all explained. You want the gory details in this paper. And so the question is then, why do these organisms then do that? Why would they want to do that? And so it really is this lifestyle trade-off between having acetate available or not in the media and being able to use all the lactate. So you can imagine here, these organisms that are recycling the acetate and able to make longer chain products, one of their main advantages is that even in acetate limiting conditions, they can use all the lactate. It's really critical, right? So if they want to grow, their main substrate is lactate, their electron donor. In order to be able to do that, they need to recycle the acetate versus directly assimilate it into products. And because of that, we see MCFA production happening. While in contrast, organisms like many of the organisms found in the gut microbiome that use butyrate and the exact same biochemistry, but seemingly only make butyrate, can't do that. So they can't recycle the acetate. They only stick to butyrate production. And you can see when they're growing in acetate limited conditions, they die off, right? So this is really a fundamental ecological trade-off that explains why organisms make medium chain fatty acids. And it really seems to be an ecological strategy of being able to maximize lactate utilization when acetate is limiting. And so these are obviously very key common metabolites present in anaerobic environments, and certainly an important thing shaping not only industrial production of MCFAs, but also what's happening in the gut itself. And yeah, you can read the gory details later. The other thing we did, again, and this is with the help of the robot, is we were able to now use our robotic capability and high throughput capability, not only to screen this trade-off on lactate, but we also looked at this trade-off in glucose and glycerol, as well as other microbes. And really, it's a fundamental thing that we saw across organisms that make MCFAs or make butyrate. So really exciting for us, people involved in MCFA production. Okay, and so the robot, as I mentioned, it was useful for getting better physiological understanding of these chain elongating microbes. The other thing I just wanted to highlight, because I know we're talking about it here, is that there's also now, we've been able to develop genetic tools to be able to move into these chain elongators. So with many anaerobes, actually, so this is very relevant for onboarding weird strains, you can't get DNA into the cell because they have what are called restriction modification systems. And so if you don't have if your plasmids, when you're doing conjugations or transformations, aren't methylated in the precise pattern of the organism's native system, the plasmid gets chewed up. And so one of the things that we've been working on is this DNA methylation platform, where we first do PacBio sequencing or nanopore sequencing to get the methylome of the organism of interest. We identify what the important methylase is, and then we'll express that methylase in our conjugal donor strain so it decorates the DNA correctly prior to doing our conjugation. And so this is just some work that we're about to publish on Clostridium clavirii, where we have our plasmid with the methylase being methylated by the native methylase and not being methylated. And so you can see when you don't have this methylation pattern, it doesn't work, you can't transform it. When you do, you can get colonies popping up and you can engineer the cells. And so we've demonstrated antibiotic resistance now in some of these chain elongators. We've also got fluorescent reporters working in a number of them. And so this is really enabling to be able to think about using this now as a platform for making a wide variety of different medium chain fatty acid products. And the starting point we've been working on quite heavily is expanding the chain length of these MCFAs. And I won't talk about that today, but if you want to bug me, you can come find me. Okay, and so we have this anaerobic platform. It's great. It's useful for getting higher phenotypic data or density. This is very much as a level 1 operation as Paul categorized it. And now the question is, how do we do this next part? How do we do this automated design of communities? And so I'll show you some preliminary data that's coming to an end here soon on that. And so to start with that, I'll take you to the tipping floor of both anaerobic digestion facilities in the city of Toronto. This particular facility we're looking at is called Dufferin Organics Processing Facility. Where you all live, where does your green bin waste go? Do you have an organics bin that then goes somewhere like a digester or a composting facility, or does it just get landfilled? Who puts their organic bin separately in a bin? Okay, so some of you, right? So in Toronto, all of our green bin waste is separated, right? So and most of it ends up at one of these two facilities, either Dufferin or Disco Roads, right? And the way it works, literally your garbage, your green bin waste gets put on the tipping floor, the truck loads it on a conveyor belt, it goes through this beautiful machine called an organics extrusion press, which is actually really critical for the technology, because what this does is it separates all the organic matter from all the grit and sand and bones, dead bodies, no, plastic bags, right? All the inorganic stuff, so you have this nice, yummy pulp, right? At least yummy for the microbes to be able to ferment, right? And so that's directly what we've been working with, including with our liquid handling robot with wide bore tips. It's about an 8% solid solution. So the plant, normally it goes to these large digesters and makes biogas. And so of course, we're in the business of converting this to other things, right? So we've now been running a bioreactor, which unfortunately I won't tell you too much about today in the interest of time, but we've been running this thing in a continuous way for over 2 years. Really, really hard work from 2 of my PhD students, Jasmine and Diana, where we have been making MCFAs first without what's called inline extraction at a yield of about 0.15 grams MCFAs per gram VS, which is pretty good yield. And when we extract the MCFAs inline, we actually have a membrane-based extraction system where we can pull the MCFAs directly out. I mentioned they're toxic, right? So when we do that, it actually increases the yield dramatically as well. We've been running that for 2 years, I said, works really well with the mixed community. And of course, now we can use that mixed community as our feeds or as our source for pulling out novel microbes, right? And so there's lots of diversity of different lactic acid bacteria, putative chain elongators. This one ended up not being actually the one we found, which is really cool. And so the question is, can we isolate these actual microbes that are growing and directly converting the food waste into MCFAs and kick out all the other ones that we don't want, right? Okay. And so one of the stars of the show that we were able to isolate just by good old-fashioned roll tube isolations and anaerobic plating is this novel strain of a Eubacterium species that remarkably converts, in this case, PY media, but also food waste to very high yields and selectivities of octanoic acid. This is very rare. All known chain elongators primarily make caporic acid, C6 over C8. This one tends to make a lot of C8, and it importantly can directly convert food waste to medium chain fatty acids, which was really exciting to us. Beyond that, and this I promise was the last biochemical pathway I'll show you, it can take lactate to MCFAs like I showed you the other microbes can, but remarkably it has an ability to, it's a very proteolytic strain. So if you look at these green bands here, you can see as it grows on peptone, as well as food waste, it rapidly accumulates ammonium. It's called a hyperammonium producing organism, and that's because it's degrading peptides. So this organism remarkably is able to degrade peptides and use those amino acids directly for chain elongation as well. What I'm showing you kind of here in the red boxes and then the green boxes, and the green boxes are amino acids that serve as electron donors into the chain elongation pathway that could also be balanced via a classic anaerobic metabolism called Stickland fermentations. And it seems like these organisms, depending on how free the amino acid source is, will either do classic Stickland fermentation, chain elongation, or some in between. And that's kind of shown on the graph here. But importantly, when it's growing on food waste in a protein form, the amino acids available, it really seems to stick to chain elongation, which is really cool and something we're still sorting out. So we can take these electron donors, and what happens is you actually get different acyl-CoAs, sometimes odd chain acyl-CoAs, sometimes branch chain acyl-CoAs, and sometimes just regular old even strain acyl-CoAs, and it can make diverse medium chain fatty acid products. So it's a really cool microbe. And what we like about it is it can take all the other types of carbon in the food waste. So there's polysaccharides, proteins, and lipids. It seems to like lactate, which is a byproduct of carbohydrate degradation, and then it can directly take the proteins and other chunks of the lipids to MCFA. So if you think about a building block for a synthetic community, it's even better than the Lactolyticus, the tooth decay microbe. So, okay, so we have this really cool microbe that one of my other PhD students, Dinesh, was able to isolate. So he has a lot of fame in the lab for finding this really cool microbe. But we wanted to find some friends, right, for this Eubacterium strain that we've isolated. And so we have this robot in our lab that's able to screen and characterize the function of microbes. In Harris Wong's lab in Colombia, they had this nice ML-driven cultivation isolation paper. And so I sent one of my students, Joel, over there. And what they have in their anaerobic chamber is just a colony picking robot. And so Joel went and spent about 4 months in the lab finding friends for the Eubacterium species. And you can see we did a whole big isolation campaign where we had regular media with antibiotics and different types of media. And then we also took actual sludge from digesters and food waste pulp and made some plates. Joel did that work at midnight because the lab really smelled really bad. And he didn't want to offend our Columbia colleagues. But even the short isolation campaign, we got really nice results, and we actually are now onboarding a very similar system into our lab currently to be able to do this all in-house. But we got this really large, diverse collection of microbes that were directly isolated from our food waste system as well as our bioreactors. And so these are organisms that are able to do medium chain fatty acid production, able to do lactate production. And so then the question is, can we use this biobank now, right, as a starting point for building synthetic consortia that even get higher titers than the isolate that we were able to find? And so the way we are doing this in general is that we first collapse this biobank into functional groups, and then that it forms the basis of our synthetic communities. So far, we've started with 4-member communities, but of course you could do any permutation of that or any combination of that. We then, this is starting from 200 strains as well. So our biobank now we have in the thousands of strains. We wanted to do this first simply, so we re-streaked and really made sure we had 200 nice starting point organisms that were diverse to work with. We then mix and match them with our liquid handling robot and directly working with the food pulp, right? So we had wide bore tips working with the food pulp. We measure their growth. Joel is a really clever guy, and he dug up some old literature from the 70s on actual MCFA production using an enzyme assay. Of course, in any of these cycles, practically, if you're using an HPLC or a GC to do your DBTL cycle, that's immediately the bottleneck, and your throughput is very, very low. So if you can do anything with a colorimetric assay and your analytics is fast, that's generally speaking going to be getting you out of that bottleneck. And then again, we use a simple model here, just a Gaussian process-based Bayesian optimization step to learn relationships between the community designs and their function or their response, and then recommend a new set of experiments. And so I have just a couple of the DBTL loops, just 2 of them to show you. Here, first, I'm showing you that this 200 strain collection we had was quite diverse. So you can see this is it growing on pulp, filtered pulp, just their media. So we can actually collect some OD measurements. You see their growth curves are diverse. And when we look at the product range, we have a functionally diverse array of 200 strains that we're also using as input for this community design. And you can see that when we look at them, they kind of cluster into some of the key functional groups we want. So here you can see organisms that are making L-lactic acid as an end product. Here are organisms making D-lactic acid. The different isomers here of lactic acid might suggest different substrate preferences of polysaccharides going into those products. We also are seeing different short-chain fatty acid producers and, of course, the MCFA producers. And so what we've done first, initially, we just ran a random cycle. We got, I think, 16 out of 860 communities that were able to produce medium chain fatty acids. And this is at quite a high titer. This is about 8 grams per liter, so already higher than our mixed, what our open mixed community culture is doing. And with the second cycle, or with the one cycle, we've really expanded the amount of communities now over 200 that are making MCFAs, right? And so now this data is going into our second cycle, which just finished today, actually, and we'll probably have our 10 cycles completed by middle of June for finalizing our community design. But already these organisms, the 4-member communities that we're getting, are able to produce higher MCFAs than just the bioreactor itself. So really exciting. So stay tuned to see what the final community looks like on that. The other thing I'll point out, that this green organism that we keep popping up, S7, that's always this Eubacterium species. So this seems really in a randomly inputted community design to be one of the key organisms for building out the synthetic consortia. The last thing we're doing, of course, when you're working with food waste is unsterile, right? And so worrying about competition, whether these communities remain stable is really key. And so we've been also doing some bioreactor trials with that Eubacterium species and some of the other synthetic communities we've been working with. And so far, it's been working good. I won't show that data now, but you can ask me about it over a beer if you're interested. Okay, so some summary points. Broadly speaking, anaerobic fermentation is really, I think, the future of chemical manufacturing, especially from all this, wide variety of food waste as a feedstock we have available. That's right now we're paying, the city pays hundreds of millions of dollars to deal with their waste. So if we can turn a revenue on that, that would be really exciting. Automation platforms are really critical for characterizing all these weird anaerobes that we don't have a lot of information of, and hopefully we can play a bit of catch up on our colleagues that work with aerobic model organisms in charting their physiology and learning about some of the rules that modulate their phenotypes. Here I showed you just that ratio of electron donor to acceptor was really key for making longer chain MCFA products, and that DNA methylation is really a critical piece of being able to actually genetically modify a lot of the anaerobes that we want to work with. And so far, the synthetic consortia that we've been building bottom up seem to be really promising for being able to recycle waste into MCFAs and then really be a platform for them bringing in our metabolic engineering tools to make the other suite of chemicals. Okay, and so of course, the next thing we're working on is trying to add this acetogenic strain into our community. So with that, I'd like to thank you guys for listening, and especially a big thanks to my team at the University of Toronto. They do all the hard work, especially Joel here in the back. He was really instrumental, one of my first PhD students, in setting up all the automation equipment. Really, really capable individual. And of course, to my collaborators here in Toronto, Columbia, and Nottingham. And with that, I'll take any questions.
A very interesting talk. So regarding the food pulp, it is just like a mechanical, I guess, you mechanically kind of, convert the food waste into these food
into
pulp.
A pulp. Yeah, let me, I can jump back if you want.
So the question is that how consistent is the composition here? Will it be like seasonal difference, I guess,
Yeah, of course. So the running joke, I guess they're helping me in the back. Get it going. There we go. So yeah, in October, right, end of Halloween season, the plants get a rush of pumpkins, right? So there's like half the waste is pumpkins. So certainly we have seasonal variability, absolutely. And so one of the main key things then is that the organisms that are selected for either in an automated way or in a rationally designed way have to be metabolically flexible. And this is kind of one of the properties that we're seeing, certainly. So we actually have a whole slew of time series data of the composition of that microbiome and how it's changing over our 2-year run. And a lot of the key players that we're seeing stick around. And when we've been analyzing their metabolism, they're quite versatile microbes. And so the Eubacterium strain we've also been feeding different batches of pulp from different years, and it remains very robust. Yeah. Unlike some of these off-the-shelf chain elongators.
Very interesting. Another quick question, like all of them are mesophiles? I mean, yeah, all the microbes. Have you been looking into compost and all those kind of places too?
Yeah, as a source, yeah, composting is generally more aerobic technology, right? But certainly you'll find some facultative anaerobes as well. Right now, we've been doing everything mesophilic because one of our main goals is to actually, we're looking at piloting some work at this facility here. And so they run at mesophilic temperatures. You can certainly do thermophilic digestion, right? Generally speaking, that's 45, 50 degrees Celsius. And certainly we could isolate and build out a biobank of thermophiles to maybe speed up the kinetics of the process as well, with the trade-off of extra energy input. Yeah.
Thank you for a very nice, interesting talk. I have a question for you. So when you have I don't know if you did it, when you have a synthetic microbiome or synthetic microbial community, how do they respond to the food? I think it's similar to the same question. Do you look at some CN ratio or something, or to
Yeah.
sure they are working?
Yes. So the amount of trace metals and nitrogen content and phosphorus and kind of macro and micronutrients typical for growth of an organism, generally speaking, are already present in the food waste. The food waste is very rich in nutrients, so we don't have an issue with needing to add any type of trace metals or anything to get the microbes that we're working with to grow in it. That being said, again, we've selected first for upfront for microbes that grow on the food waste. When we take any of the chain elongators that we have that we're not isolated on our food waste, they don't grow on it. So it's a critical step in getting organisms to actually work on complex feedstocks. Yeah.
Great talk, Chris.
Thank you.
I was thinking about the scalability of the process, meaning that if I understood well, when you are doing high throughput, you utilize only the soluble fraction, you don't utilize solids, so then you can measure growth. So then when you go to a bioreactor where you have solids and hydrolysis, how reproducible it is? Like
No, so we do work with solids. Yeah, we work with the pulp directly.
With the solids in the plates?
Yep. So if we use wide bore tips and we shake the pulp and get it homogenized, and we're directly pipetting the pulp. And we've done a bunch of controls and things to normalize. We have positive controls, negative controls. In our last DBTL cycles, which took a lot of little time to sort out how to get it to be reproducible, we're working directly with the pulp.
Oh, cool.
Yeah.
Okay, I missed that.
Yeah.
Yeah, thanks for a great talk. I'm curious, you mentioned that the methylation prep for DNA plasmids works with many microbes in this case. What would you say the percentage of the ones that you've tried work?
We haven't tried tons. I know folks at Oak Ridge definitely are the experts, so I talked to Carrie on that. They've tried a lot more. Nigel Minchin's group's also tried quite a lot. We have tried it with a couple, and it works. But across the board for almost all the anaerobes we work with, if you just try to do conjugation or transformation, it doesn't work. They won't grow. So it seems anecdotally anyways that this is a critical step. And again, folks at Oak Ridge are the experts in that. Yeah.
Really good stuff, Chris. Do you utilize any modeling approaches to simulate carbon energy partitioning across your communities and
big time? Yeah. And so actually, I of course I didn't get into the weeds because I ran out of time, but you can certainly go look at the preprint. But all of the work here, and of course, I exited, so that's going to be gone. But I'm sure they'll save me. All of the work for sorting out, this lifestyle trade-off between butyrate and MCFA producers was all anchored in simple metabolic models, but more importantly, a basic metabolic model that's just stoichiometric isn't going to work for anaerobes, I think. You also need to layer on resource allocation to account for protein costs, because as you can see here, which I didn't get into the details of, many of the decisions, phenotypic decisions that organisms are making are based on protein investment. And so here, this is a key one, of course. So we used, in this case, enzyme cost minimization to calculate enzyme costs, which is a technique that came out of, it's a guy's name. Escape me for now, but there's lots of good work on that. But yeah, so resource allocation modeling based on simple core models of metabolism is generally what we use. If we use a very complex full-scale genome scale model, oftentimes we run into issues with false reactions or promiscuous or spurious reactions. So we try to build simple core metabolic models and layer on resource allocation type of constraints. Absolutely essential, I think, if you're going to do any type of metabolic modeling with anaerobes. Yeah.
Thanks, Chris. That was a really cool talk. I was curious, in running your DBTL loops, what's been your biggest bottleneck? Like, what's preventing you from running the loop faster?
The microbes, they just grow too slow, right? So anaerobes, of course, their doubling time is 24 hours, something like this in the hours range. And so our cycles, practically speaking, take a week, right? So we start pre-cultures, get all of our D96 plates ready to roll from our biobanks, and then when we do our incubations, they take 7 days. And so that's the limitation. We can reduce our time maybe to something like 4 or 5 days, not fully capturing the final titer, but capturing how quick the organisms can make the MC phase, and that might actually better capture rates into our objective function for a DBTL versus just titer. But that's something we haven't started to play with yet. Yeah, but just the time the microbes take to grow.
When you're measuring titer, are you just measuring at the final point? Okay.
Yeah. So back to Davina's question about a mixed pulp. Of course, if we had a nice, soluble, clear media, we could be measuring growth and all those things kind of dynamically. And certainly, actually, when we started this, we were doing that. We were using just the soluble fraction, but then we changed course because we thought, to your point, Davina, it'd be much more impactful to try to do this directly with the pulp. Yeah. So we actually have more DBTL cycles on just soluble media, but we've switched recently to the pulp. Yeah.
Any more questions? Oh, Kim.
I feel like this is getting into the weeds a little bit, but in terms of separating out the supernatant from the pulp that you're talking about, what are you using, and is that integrated into that?
Yeah, we don't have a centrifuge here, so we do that offline. So after the fermentation completes, we then take the plates out, spin them down, and do our enzyme assay. Of course, you could also do HPLC or GC, but again, it's very slow. So if we were going to do this cycle with GC, I think our runtime would be like half a year at least already on GC time, on a quick GC method, like an 8-minute GC method on just already the data that we've collected. So really important consideration.